Hello, and thank you to ARM and the Meeting on the Mesa folks for the opportunity to represent my colleagues at Genocea, where we are developing what we hope to be next generation neoantigen targeted solid tumor T-cell therapies. I'll of course be making forward-looking statements, and so I ask you to pay attention to this disclaimer briefly. But let's focus on the Atlas and Genocea story. We think what sets us apart is a focus on neoantigen selection. Our platform, Atlas, is a differentiated antigen discovery platform for which we think we already have substantial human proof of concept in the form of best-in-class immunogenicity and clinical efficacy data from our neoantigen vaccine, which we call Gen9. And armed with that proof of concept, we are in the midst of a clinical trial looking to treat checkpoint refractory patients with our solid tumor neoantigen targeted adoptive T-cell therapy, which we call Gen11, for which we expect to have initial human data in the window of late Q4 through Q1 of next year. So let me talk about our Atlas platform and why we firmly believe that better antigen selection is a critical thing to drive better efficacy in cell therapies. With our Atlas platform, we are able to use patient-specific biology, not predictions, to identify and prioritize all surface-presented antigens that we want to be targeting with our T cells. We achieve this as described in this picture below, where we're collecting some PBMCs from the patient as well as a tumor sample, which could be an archived sample. We send that for next generation sequencing and get a list of all of the candidate neoantigens, which could be not just point mutations, but also indels, frame shifts, gene fusions, even viral antigens. And we take each candidate and we clone them separately into bacterial vectors that we've engineered to be taken up by antigen presenting cells and drive presentation either through MHC class 1 for CD8 T cell responses or class 2 for CD4s. So using the patient's own antigen presenting cells, which we derive from the PBMCs, we feed every antigen separately using these vectors into those cells. We expose the cells either to the CD8s or CD4s from that patient. And the output we depict on the right, where we can array the immune responses as measured by cytokines such as interferon gamma and TNF alpha that the patient makes to every candidate neoantigen. And we're looking for those antigens that pop from background, that drive strong anti-tumor cytokine responses. Those antigens are the ones for which we expect to expand T cells to drive compelling therapeutic benefit, we hope. We're also ruling out these critical antigens that only Genocea can identify. We call these inhibigens that actively suppress the anti-tumor cytokine responses. We make sure not to include such antigens, which are often misidentified and mischaracterized by algorithms, and ensure that our final product is highly neoantigen specific, and not just that, but specific for the neoantigens that drive anti-tumor responses in the patient, which we hope to amplify. The human proof of concept for this platform comes in the form of the clinical data that I've hinted at with our Gen9 neoantigen vaccine. These are immunogenicity data from the first in-human study we conducted there. And what you can see is that across the 8 patients that we dosed with monotherapy to Gen9, we detected amplified immune responses to all but one of the more than 100 antigens across those 8 patients that we prioritized in our vaccine. We contrast that with the immune response frequencies that our peers have shown in similar trial settings. And all of them, seemingly because they are working from algorithms, drive about 60% immune response frequency, meaning about 40% of these vaccine approaches have been junk. These data are very exciting and were the reason these data were a top 10 immuno-oncology abstract at ASCO in a couple of years ago. We've subsequently shown that this compelling immune response drives clinical benefit. And what you're seeing here is data from our dosing in combination with checkpoint inhibitor, where most intriguingly, in several patients who had been on checkpoint inhibitor for about 4 months while we made the vaccine, saw post-vaccination with Gen9 separate, independent RECIST score tumor responses, seemingly driven by the Gen9 vaccine. The graph on the left shows you the overall time. The graph on the right shows you the tumor response curves after vaccination. But this frequency of response in patients who had already achieved stability on checkpoint inhibitors, I think is a compelling additional data point suggesting that our strong immune response frequency and magnitude leads to clinical benefit in patients. And we're using these insights at the heart of our Gen11 neoantigen targeted T-cell therapy. We call it an NPT because not only are we targeting at neoantigens the right way, but we're using peripheral blood. And I'll speak to the advantages of both sides of the equation now. We think of this as a third category of neoantigen targeted cell therapies because not only do we benefit from Atlas to choose the right antigens, but then we benefit from our Planet manufacturing process where we take peripheral blood from the patient and preferentially expand only those T cells to these Atlas prioritized neoantigens. And so the resulting product, Gen11, we call an NPT, is non-engineered and still positioned to provide patient benefits in efficacy, accessibility, and scalability through better T cells to better targets. The better T cell point, I think, is also underappreciated, where I think there are significant challenges associated with tumor infiltrating lymphocytes or TILs. They're hard to access, whereas of course, with peripheral blood, we simply need to do a blood draw from the arm. Others have also demonstrated that these T cells are younger and fresher, if you will, and have a greater potential, therefore, for proliferation and persistence. And some of the evidence confirming this comes in the form of responses to checkpoint inhibitor therapy, where it has been shown that it's not simply TILs, but actually shock troops, if you will, from the periphery that home to the tumor and drive lasting clinical benefit. So all of these facts point to the potential benefits of NPTs and derived from peripheral blood, which is core to our product. We expand these peripheral blood-derived T cells through our Planet process, which we've designed to be highly scalable and rapidly optimizable. And so as we advance from this initial proof of concept study, we expect to be able to achieve an overall needle-to-needle time of between 6 and 8 weeks at commercial scale, which we think is going to be very compelling in the clinical setting, especially when one considers the potential efficacy and safety and accessibility advantages of Gen11. So overall, we think of the NPT as a third and differentiated category of neoantigen targeted T-cell therapies, in contrast to TILs, which have shown compelling efficacy in certain patients, but suffer from the challenges of the cost and inconvenience of the ways in which TIL extraction is performed, suffer from the fact that much of the final product is not even tumor specific, which may have implications for efficacy, certainty of response, as well as for product release. And finally, it may be that these TILs are actually an exhausted phenotype, and therefore the durability of response in some patients may be of question. And while TCRTs may have some advantages that contrast with the TIL disadvantages, it's also the case that TCRTs are HLA restricted. And so essentially patients have to be of a homogeneous subset of Western Europeans to have optimal benefit from TCR therapy. And it's, I think, just as significantly the case that TCRs target just a small handful of antigens in the patient and with only preferentially CD8 T cells. And so being able to go after a breadth of tumor targets with both CD8 and CD4s, we think is a way to improve. And so our NPTs have advantages over both of these approaches. We're in the midst of a clinical study, what we call the TITAN study, where we are dosing a basket of patients in CPI-resistant solid tumors. The patients are in two cohorts. What we call cohort B would be the conventional TIL regimen cohort, where patients have standard Flu/Cy lymphodepletion prior to Gen11 dosing, and then daily high-dose IL-2 subsequently. We also have what we call cohort A, where patients are not lymphodepleted, receive a substantially reduced dose of IL-2, and instead of getting Gen11 as a single dose, receive portions of it every 3 weeks over time. This two-pronged approach gives us both a nice way to contrast and compare our results with standard therapy through cohort B, as well as a way to potentially really open up the accessibility of our therapy to the community setting by avoiding lymphodepletion and potentially even hospitalization through the intermediate dose of IL-2. We're very excited to be able to report on the safety, on biomarkers of persistence and proliferation, and of course on initial clinical efficacy in a subset of these patients, starting in this window of late Q4 through Q1. So in sum, we think of Gen11 as really potentially transforming neoantigen T-cell therapy for solid tumors. The benefits come from Atlas, where we are able to prioritize the right T cells, CD8 and CD4 T cells, to the right surface presented neoantigens that drive anti-tumor responses, targeting a breadth of up to 30 neoantigens in our final product. We use peripheral blood, which has the advantage of greater accessibility, activity, and durability, all of which we bring together in our Planet process, which we've designed with the commercial target in mind. So for all of these reasons, we believe that Gen11 may prove to be a highly exciting next generation neoantigen targeted cell therapy. And so I'll just remind you again that all of this starts from the neoantigen selection process built on our Atlas platform, confirmed in humans through our Gen9 neoantigen targeted vaccine, and positioning our Gen11 for what we hope to be an exciting clinical readout starting very soon. So I thank ARM for their opportunity to present, and I thank you for your attention.